This presentation is on the effects of diet and lighting condition on the Nile grass rat. Our research question is, will a high-fat diet and lighting intensity affect anxiety, risk-taking, and cognition in the Nile grass rats? Statistics show that the leading epidemic in the world right now is obesity, which is most often caused by a high-fat diet in combination with other factors. With obesity often comes some underlying diseases such as diabetes, cancer, cardiovascular disease, depression, and anxiety. This doesn't just affect adults. It affects people from all populations. 1 in 5 kids in the U.S. are obese, and a total of 93.3 million U.S. adults are obese. A link between high-fat diets and obesity has long been established. However, the connection between high-fat diets and mental state is still under investigation. Previous research has shown that high-fat diets impair neural function, both in relation to mental health and cognitive functions such as memory and spatial tasks. We are also investigating the effects of lighting condition on mood and cognition. A relationship between dim lighting and depression, which is often associated with anxiety, is obvious in seasonal affective disorder in humans. But there are also other cognitive effects of lighting that are being researched. We hope to expand the knowledge base on the effects of a high-fat diet and lighting on mood and cognition with the following research.
In this study, we use Nile grass rats as the subjects because they are diurnal, meaning that they sleep at night. The importance of using a diurnal rodent is clear because their circadian rhythms are similar to those of humans. That means that any significant results gathered from this experiment will be more generalizable to humans, which in the end is the goal of the study. So in total, 27 rats were tested in this experiment. 13 of these were male and 14 were female. Of course, no animals were harmed in the process of this experiment, although there were some close calls during the Morris water maze.
The grass rats were singly housed and allowed to acclimate to this condition for 1 week. They were then randomly assigned into one of the following conditions. 8 rats were assigned to the control food and control lights conditions. 8 rats were assigned to the high-fat diet and control lights condition. 5 rats were assigned to the control food and dim lights condition, and 5 rats were assigned to the high-fat diet and dim lights condition. The animals were in these conditions for 6 weeks before behavioral testing began. The control food
was regular lab food, and the high-fat diet was Ruffles, Jif peanut butter, Chips Ahoy, Nesquik, and Rat Chow.
The following 3 weeks, each lab group ran the 2 animals assigned to them in a series of behavioral tests. The first week, we ran the rats on a test called the open field test, and the second week, we ran a test called the elevated plus maze, and in the third week, we ran a test called the Morris water maze. In addition, the mass of all the rats was recorded on a weekly basis, and Dr. Gall also examined the rats' circadian rhythms after the 3 weeks of testing.
So our hypothesis for this study was a high-fat diet and lighting intensity will affect anxiety-like behaviors, risk-taking behaviors, and cognition in a diurnal rodent model, the Nile grass rat. So we hypothesized that these 2 conditions would result in increased anxiety, decreased risk-taking behaviors, and decreased cognitive functioning, with the most impactful conditioning being the high-fat diet and dim lighting conditions.
So the first week, as previously mentioned, was the open field test. Our materials were the open field box, the Nile grass rat, and the software system, which was the AnyMaze behavioral tracking software, which collected data from every single test. During the first week of testing, each rat went through the open field test, where each rat was placed one at a time. The rat was then released from the net and placed in the center of the plywood box. The rat was in the open field for 5 minutes as researchers noted the rat's behavior. At the end of the 5-minute trial, rats were returned to their cages. We looked at the entries into center, entries into perimeter, distance in the center, distance in perimeter, time in center, and time in perimeter. And then time spent in the perimeters of the open field test would show that there was a higher level of anxiety.
So the materials used for the elevated plus maze was obviously the box, the Nile grass rat, and the software system. So for this procedure, as before, each rat was tested on each one time. The researcher placed the rat into the center of the maze with its head facing out to one of the open arms. The rat was in the maze for 5 minutes while both researchers and the video tracking system recorded the rat's behavior. At the end of the 5 minutes, the video tracking system stopped recording and researchers returned the rat to its cage. The elevated plus maze tested anxiety and risk-taking behaviors. For the measures, the test measures measured entries into closed arms, entries into open arms, distance in closed arms, distance in open arms, time in the closed arms, and time in the open arms. The rats that spent more time or made more entries into the closed arms of the maze exhibited anxiety behaviors through their preference for the more comfortable enclosed area. The time on the open arms of the maze was also considered risky behavior, as they were more likely to fall into the water from these arms. That being said, the time spent in the closed arms exhibited less risky behavior.
The Morris water maze was used to assess the rat's spatial memory and learning ability. The main materials we used for this were a tub for water, a hidden platform below the waterline, and 8 visual cues placed along the walls of the tub. The water contained a low concentration of white tempera paint to make it opaque, thereby hiding the platform from the rat's view. Each rat was first placed onto the platform for 15 seconds to allow it to get a sense of where the platform was in the tub. They were then placed into the water on the side of the tub opposite from the platform and were given 60 seconds to find the platform while the video tracking system recorded their movement. If a rat didn't find the platform within that minute, it was placed back onto the platform for 15 more seconds before being returned to its cage. If they did find it within the minute, they were placed back into their cage to rest before the next trial. Each rat ran 4 trials on the maze to see whether the amount of time taken to find the platform decreased in each successive trial. We measured the amount of time taken to reach the platform, if they reached the platform, as well as the amount of time spent in the quadrant of the tub that contained the platform. Our group had some issues because our rats didn't exactly want to swim, but in theory this test is good for assessing cognitive functions like spatial memory and learning ability.
The results of the open field test showed that rats in the high-fat diet and bright lights condition spent less time in the center than the rats in the controlled diet and bright light condition. The results for the elevated plus maze showed that the rats in the high-fat diet condition traveled less distance overall and also entered the center of the maze less. The results of the Morris water maze showed no significant effect of lighting or diet, but they did show a trend that dim lighting decreased the time taken to reach the platform. The results of these tests showed that the rats who were fed a high-fat diet and kept in dim lighting were more likely to have anxiety and less likely to exhibit risk-taking behaviors. There were no significant effects of high-fat diet and dim lighting on cognition, therefore the hypotheses of this study were only partially supported.
So the figure on the left showed that the differences in the average amount of time spent in the center of the open field by which condition the rats were in. Diet and lighting were shown to have a significant interaction effect on the amount of time spent in the center, such that the rats raised in the control diet and bright light conditions, presumably the least anxious rats, spent the most time in the center of the open field. This indicates that higher anxiety levels were caused by an interaction of diet and lighting condition. So the figure on the right shows a significant main effect of lighting on the number of entries to the center, such that rats living in bright light entered the center more often than rats in the dim light. In conclusion, the bright light condition correlated with more entries into the center of the elevated plus maze. Again, the figure on the left showed that there were no significant effects recorded from this test. The graph, however, depicts a trend that showed the rats in dim lighting may have taken a shorter amount of time to reach the platform in comparison to those in the bright lighting. In conclusion, although Morris water maze did not yield any significant results, further research may produce different results. And finally, the figure on the right showed that there was a significant difference in the mean weights of the rats fed the high-fat diet and the rats fed the controlled diet. The graph shows that the rats on the controlled diet weighed significantly less than the rats with the high-fat diet. So in conclusion, a high-fat diet did cause a significantly higher body weight in the Nile grass rat.
The purpose of the following study was to examine whether or not a high-fat diet and dim lighting would affect on anxiety-like behaviors, risk-taking behaviors, and cognition in the diurnal rodent model, the Nile grass rat. Similar research has been done which has found similar results to ours, which help us validate our study even more. As with any experiment, there are some limitations to the study. The number of Nile grass rats we had could be a limitation due to the sample size. There could also be a limitation in the equipment we used or simply human error. There are several strengths to our study. One strength is the fact that our subjects were the Nile grass rats, which can allow for results to potentially apply to humans, which is ultimately the goal of the study. Another strength of our study is that it was an overall well-controlled experiment. Rats were tested at the same time each week, and the variables other than the independent variables were constantly held the same. Future research can be done off of our research. Studies can manipulate the length of time in each lighting or could alternate the size of the cage the animals are housed in. Researchers could also use a maze that does not involve water but is solely done in dry conditions. Ultimately, this study gave us exciting insights on links between obesity and mood conditions. By looking at how these behaviors in the Nile grass rat were affected, we can greater assess the implications of diet and lighting conditions in human beings, specifically with obesity. According to this research, the same diet that causes obesity may also be inducing anxiety. This can also be flipped, as anxiety can alter a person's metabolism, making them obese. As for dim lighting, one can see the effects of seasonal depression, and other effects of lighting on mood are under investigation. We would like to thank Dr. Gohl for allowing us to have this experiment. We also want to thank the Hope College Neuroscience Department and the Hope College Institutional Animal Care and Use of Committee.